So there are different types of amyloid signature proteins, and I feel like I'm qualified to talk about one specific type, and these are called apolipoproteins because we worked for many years with these proteins in the context of lipid transport. And also these are essential proteins which always co-deposit with various amyloids regardless of the fibril-forming protein. So they are always found in deposits in non-fibrillar form, and it is not entirely clear what they are doing there and why they co-deposit, but they are used to help diagnostics. So essentially, my understanding is that these proteins are unique, have unique structural and functional properties. They are highly flexible, and they are comprised of amphipathic alpha helices, which means alpha helices which have one hydrophobic phase and one very charged and hydrophilic place. So they tend to bind to hydrophobic surfaces. And such surfaces are always present in all patient-derived amyloids due to their molecular packing, where the molecules are packed in stacks, flattened molecules packed in stacks, one on the top of the other. So if you have a hydrophobic residue in molecule 1, it's sitting right on the top of an identical residue in molecule 3, 4, and so forth. So what we have revealed in our computational studies, but also based on our knowledge of how these proteins behave experimentally, is that these proteins open up on amyloid to expose their hydrophobic phases, which are normally sequestered from the solvent, and then they spread along the amyloid surface to cover this hydrophobic phase so that it is now covered with amphiphilic surface, with mostly charged surface, and thereby they stabilize amyloids. The question, going back to your original question, so how does it affect the properties of amyloids? And what is the role of these proteins? So we understand, we think how they bind. Again, it's at this point, it hasn't been directly proven by experiment, but there are tons and tons of indirect experimental evidence to support this binding mode. And it's not limited to one particular apolipoprotein and one particular amyloid, but it extends. We think it's pretty general and common. So the question now is, how do these proteins influence the properties of amyloids? And if you can imagine, you have a tree with a vine growing up the stalk, along the stalk, then would it influence the tree a lot? We don't know. We think that by binding to the tree, this vine prevents other hydrophobic molecules, such as lipids, from binding to the same site. We also think that the important thing is how this particular amyloid propagates. Some amyloids propagate through fragmentation, when a long fibril breaks into small fragments, and these fragments move away and generate nuclei for new fibrils to form. I would expect that apolipoproteins, by binding along the fibril, it slow down this process, stabilize against breaking the fibrils. At the same time, other amyloids propagate through so-called secondary nucleation. That is that a new nucleus is formed along the side of the fibril and then separates from it and goes off on its merry way. And here, we don't know how the amyloid, how the bound apolipoproteins will influence the process. It really depends whether the nucleus is budding of the hydrophobic site, then it will be blocked, or if it is budding of the hydrophilic site, then there might be no effect. Another important thing is that we realized how paramount is the amyloid polymorphism to these kinds of interactions. And that is a factor that was previously unappreciated or perhaps underappreciated, because especially for small peptides, many proteins and peptides tend to form various amyloid structures, not a single fold, but multiple folds. And as a result, the surface of the amyloid fibril in these different structural polymorphs will be very different. And in some fibrils, amyloid signature proteins such as apolipoproteins may bind at the fibril ends. And in other fibrils, they may bind along the fibril surface. So the effects will be different. For instance, if amyloid growth is very important for amyloid propagation, if elongation of the fibril is important, and then it breaks off, then binding of apolipoprotein at the top or bottom will block this process. So there the effect is obvious. So the specifics of the binding depend, of course, upon the fibrillar protein, but not only the protein, the fiber structure, which might be different for the same protein in different patients, for example. So, and of course, in the in vitro studies that lots of people are carrying out, myself including, most of the time we don't know what the structure is. And this leads to a lot of controversy in the field because the binding sites will be different. But generally, we understand why apolipoproteins co-deposit with amyloids, because they both have extended long hydrophobic surfaces, which, if open to the solvent, are energetically unfavorable, so they tend to stick to each other. But there is no simple answer to your question, what is the effect of such binding on the biological properties? It is a function of amyloid structure, first and foremost. So more studies are needed.